Sejal, hi. Sejal Tondwalker here. Uh, I have today we are going to see the third part of the chapter properties of inheritance and variation. Uh, um, in the last video, we completed till dihybrid cross. Today, we'll start further, that is chromosomal theory of inheritance. Chromosomal theory of inheritance. Chromosomal theory of inheritance. It was proposed by Walter Sutton and Theodore Boveri in 1902. It states that pairing or separation of pair of chromosomes leads to segregation of a pair of factors they carry. Sutton united chromosomal segregation with Mendelian principles and called it chromosomal theory of inheritance. Important statements regarding chromosomal theory of inheritance. Chromosomes are vehicles of heredity. Means chromosomes contains genes and genes are the basic fundamental units. So chromosomes act as a carrier. They are carriers. They are transferred from parents to offspring. Chromosomes are transferred from parents to offspring. Chromosome contains gene, that is chromosome act as a carrier for genes, that is they are immortal. Two identical chromosomes with similar genes form a homologous pair. Homologous pair segregates during gamete formation of anaphase 1 of meiosis 1. Independent pair segregates independently of each other. Chromosomes are mutable. Genes are present on the chromosome, hence show similar behavior. Morgan's experiment. Thomas Hunt Morgan worked with fruit flies, also known as Drosophila, to prove chromosomal theory of inheritance. Why Drosophila is suitable for this genetic study of Morgan's. So basically, the reasons for Drosophila being suitable are similar to the P being suitable for Mendel's experiment. So let's go through them. Drosophila breeds very quickly, hence the outcome will be earlier. Short generation time, they have life cycle of about 12 to 14 days. Their breeding can be done throughout the year. That is, they do not require specific season to be bred. Hundreds of progenies per mating. That is, in one reproduction or in one mating, we can get hundreds of progenies. They can grow on simple synthetic medium. Male and female flies are easily distinguishable. It has many hereditary variations that can be seen with low per microscopes. Chromosomes of Drosophila are shown below. Male has X and Y, and female has X and X chromosome. Linkage and recombination. Linkage. What is linkage? Linkage is physical association of two or more genes on a chromosome. Linked genes do not show independent assortment. What is recombination? Recombination is generation of non-parental gene combination. Recombination occurs at the prophase pachytime stage of prophase 1 of meiosis 1. So here there are two chromosomes. Here the distance between A and B Here A, B, and C are dominant. A, capital small A, B, and C are recessive. But the distance between two genes, that is A and B, is small. Hence, they are known as tightly linked. In case of this one, the distance between A and B is wide. Hence, they are known as loosely linked. Now, actually, according to genetic study, loosely linked is preferred over tightly linked, as loosely linked produce more recombination. So, here a loosely linked pair is taken, which is crossed. Cross means एक की टोपी दूसरे like this is small c, which is actually part of this, is now becoming a part of this due to recombination. Morgan carried out several dihybrid crosses in Drosophila to study sex-linked genes. We'll go through these crosses. Morgan carried out two dihybrid crosses. So we'll first see about cross A and then cross B. Both the crosses are almost same, just the parental genes are different. So cross one. For cross one, yellow-bodied, white-eyed female, which is recessive. He took yellow-bodied, white-eyed female. Its genotype is small y, small y, small w, small w. And in case of male, he took brown-bodied, red-eyed males, which is a wild type. With wild type here basically means dominant. Moreover, uh, the Morgan's experiment differ from Mendel's because here he took dominant genotype as not capital letter of the same, but instead of capital letter he took plus signs. See here. Here for brown-bodied, red-eyed males, he took Y plus, Y plus, W plus, W plus. So that is the difference between Mendel and Morgan's experiment. In case of cross 2, he took white-eyed miniature wing. Miniature means small winged. Its genotype is WWMM. This is a recessive female. And in case of male, wild type, he took wild type, that is dominant, red-eyed and large wing. Its genotype will be W plus, W plus, M plus, M plus. So now let's go through the diagrammatic representation of Mendel's of Morgan's cross given below. So here it is. First, we'll go through the cross A. Let's see what happens in cross A. Here in cross A, first this is recessive female, that is YWYW, and this is dominant or wild type male, Y plus W plus. The another, this represents Y chromosome, as we have seen. Drosophila male has X and Y chromosome. So now what will happen is these Y plus YW will first go with Y plus W plus and then with wild type. Okay, so we got this. When this goes with this, we got wild type, and when YW goes with Y, we got recessive, that is yellow white. Now, in F in F2 generation, what happens? These go both parental and recombination. So first we'll see for parental and then for recombination. Okay. Now, what happens in parental is, as we have seen earlier, that is, these YW first goes with YW to form yellow white. These YW then go with Y to form to form YW and Y, that is yellow white. Next is these Y plus and W plus goes with YW to form this Y type, and go with Y to form this. Okay, so basically this is parental. Parental is easier to understand. Now, what about recombination? So, as we have studied earlier, recombination means uski to bhi uske saath. So basically what happens, let's see. These two recombines. Which two? These two have recombination. Therefore, what do we get? What do we get is this is Y and this is W. Next, we got us this, which is Y plus, and this, which is W plus. Now, basically, what happens is this goes with Y and W. So Y W plus goes with Y and W to form this yellow. Now again this Y W plus goes with Y chromosome to form this. This Y plus W now goes with Y and W to form white. And this goes with Y to form understood. So basically, now this is recombination. We saw what happens in parental and how recombination takes place too. The parental percentage is 98.7% in cross A, and it is 1.3% recombinant person. Okay. So if you compare this with cross B, you will find a great difference between the percentages. See, here the parental type percent was 98.7, but in case of crossbreed it is 62.8%. And what about recombinant percent? It was 1.3% in cross C and 37.2% in cross B. So why so large difference? So the cause of this difference is none other than other thing, but as you've seen in crossing, the distance between the Y plus or W plus or Y and W is not much larger. They are tightly packed. We can say that. But in case of cross B, the distance between W and NG or W plus and M plus gene is quite large. And we can say that they are loosely packed. And we know recombination is favored by loosely packed chromosomes. Therefore, the recombinant percentage is high in cross B. Now we move further. Now we move further to polygenic inheritance. We move to polygenic inheritance. They are also known as non-Mendelians. Now let's see what is polygenic inheritance. It is the inheritance. It is the inheritance in which some traits are controlled by 3 or more genes or multiple genes. Basically, one trait is controlled by more than 3 genes. Examples are given, that is human height and human skin color. So it also takes into account the influence of environment. So here it basically says that our height, we have seen that we know that tall is dominant and dwarf is recessive. But have you, are every people present here either tall or either dwarf? No, some have intermediate height. So why intermediate height? Because there is more than more than 3 genes involved here. So they have intermediate height. Same goes to skin color. We have light skin tone, we have dark skin tone, we have intermediate skin tone too. So it also takes into account the influence of environment. In the phenotype, reflects the contribution of each allele. That is, each allele contributes a certain result in the phenotype. Okay. Now we'll study about human skin color. It is mentioned in NCERT. So let's see. But what do we mean by this phenotype reflects the contribution in polygenic inheritance? The effect of each allele is added. That is, it is additive. Now we'll study about human skin color. So human skin color, human skin color is controlled by 3 genes. We assume them as A, B, and C. They control our human skin color. If all here ABC, capital ABC, is dominant form, which represents dark skin, and this is the form of small ABC, which represents light skin. If the genotype is capital A, capital A, capital B, capital B, capital C, then it is the phenotype is the darkest skin color. And if all these 6 alleles are or 6 genes are recessive, it has the lightest skin color. But what if there are 3 dominant and 3 recessive? Then it has intermediate skin color. Going further, now it is pleiotropy. What is pleiotropy? Pleiotropy. Your single gene exhibits multiple phenotypic expression. Such genes are called pleiotropic gene. That is, a single gene exhibit multiple, that is more than one character. Like we know, gene for height exhibits only about height, not a skin color. But there are certain genes which exhibits more than one thing. So they are known as Cleotropic Gene. Now, in most cases, mechanism of pleiotropy is the effect of a gene on metabolic pathway which is contributes towards the different phenotype. However, in most cases, mechanism of pleiotropy, that is, when gene affect our metabolic pathway, a pleiotropy occurs. Example of pleiotropies are given here. That is starch synthesizes in P and phenylketonuria and sickle cell anemia in humans. We'll study more in detail about these two disorders in later chapter, further in this chapter only. But here short is given. Phenylketonuria and sickle cell anemia are genetic disease in which the mutant gene exhibits many phenotypic effects. Example, in phenylketonuria results in mental retardation, reduction in hair, and skin pigmentation. That is a gene which causes phenylketonuria, also cause mental retardation, reduction in hair, and skin pigmentation in body. Let's go further. That is starch synthesizes in pea plant, which is an example of, you know, pleiotropic. So, starch synthesizes effect is effectively by BB, capital BB. Therefore, large starch grains are produced. If the with this, a small BB have less starch synthesis efficiency and therefore produce smaller starch grains. It also shows incomplete dominance. Here we can see that here it is capital B's, that is it has higher starch synthesis efficiency. There it will also has large seed size. And here small BB, it has less starch efficiency production or synthesis efficiency and will have small size seed. Now on mating this, we get a starch grain with intermediate seed size. So this is also an example of incomplete dominance. Now on moving further, we came to a new topic, that is sex determination. We all know that humans have 46 chromosomes and 23 pegs, that is, out of which human chromosomes are divided into autosomes and sex chromosomes. Now, what are autosomes? Autosomes contain somatic character, that is, they are responsible for our body growth and all. First, 22 pairs are known as autosomes. Sex chromosome. Sex chromosome controls our gender. They specify our gender. 23rd pair of a chromosome is sex chromosome. If the sex chromosome is XX, then the individual is female. And if it is XY, then the individual is male. Autosomes are chromosomes other than sex chromosome. Autosome number is same in male and female in humans. That is, male also have 44 autosomes, female to have 44 autosomes. Sex chromosome X and Y determine the sex of the individual. That is, if the progeny or offspring got two X chromosome, then that progeny is female. But if it got one X chromosome and one Y chromosome, it is a male progeny. Hanking in 1891 studied spermatogenesis in some insects and discovered the X chromosome. So, Hanking discovered the X chromosome. Now we'll see mechanism of sex distribution. First one is XX-XO mechanism. In this, male is heterogametic, that is, both the gamete produced are different. That is, it has chromosome XO, 6 chromosome XO. Here, half of the gametes will have X chromosome in them, and half of the gametes will have O chromosome, no X chromosome in them. This O is basically not O, it is 0, but we say it as O. So, in case of males, in XX-XO mechanism animal, the half of the gametes will have X chromosome and half without X chromosome. In case of females, they are homogametic, that is XX, that therefore all the gametes produced by females will have X chromosome in them. Example of this mechanism is grasshopper and certain insects too. Now we got XX-XY mechanism. In XX-XY mechanism, in case of male, male is heterogametic. Same, the gametes will be different. The chromosome is X and Y. Therefore, half of the gametes produced by male will have X chromosome and half of them will have Y chromosome. Here, the difference between XXXO and XXXY is here only X chromosome was present in case of males, but here Y chromosome is also present. In case of XXXY, females are homogametic, that is XX, therefore all the gametes produced will have X chromosome. Examples of XX and XY mechanism are human beings and Drosophila. Now, third mechanism is ZZ-ZW mechanism. ZZ and ZW mechanism is quite different from XX-XO and XX-XY mechanisms. How? We'll see now. In this ZZ-ZYW mechanism, males are homogametic. Males are homogametic. That is, they have similar gametes. Therefore, all the gametes produced by the males will have Z chromosome. But what about females? Females are heterogametic. That is ZW. Therefore, half of the gametes will have Z chromosome and half W. So, here's this mechanism is followed by births. So here's the difference with that is XX, XY or XX and X naught mechanism shows male heterogamecy, that is males are heterogamic, but ZZ-ZW shows female heterogamecy, that is females are heterogametes. Yeah. See, here it is given in a diagrammatic representation. The humans, Drosophila, and birds. So in case of human, we have XY and XX. Drosophila to have X1, XX. Both will have ZZ. Males will have ZZ and females will have ZW. Okay. Now moving further, let's study sex determination in humans. So, sex determination in humans. We know humans has 23 pairs of chromosome and 46 chromosomes. We also know that X represent female. X is present in male too and female too, but there is one additional X in. Sorry, females have XX sex chromosome and males have XY sex chromosome. Now, during spermatogenesis, males produce two types of gamete. As they are heterogamete, they produces two types of gametes: 50% with X chromosome and 50% with Y chromosome. We have studied this earlier too. And whereas female produce only one type of ovum with X chromosome. Okay. There is an equal probability of fertilization of ovum with this sperm carrying either X or Y chromosome. That is, they are produced in the ratio of 50-50, that is 1:1. There are 50% chances of X sperm carrying X chromosome to fertilize the ovum, and there are also 50% chances of sperm carrying Y chromosome to fertilize the ovum. Therefore, sperm determines the gender of a progeny. Now we'll see sex determination in honeybee. Sex determination in honeybee. It is based on the number of sets of chromosome individual receives. Sex determination in honeybee depends on the number of chromosomes an individual receives. We'll study this in detail. An offspring's year in this system, males produce sperms by mitosis. They do not have father. We'll see them. See, your females have 32 chromosomes. How many? 32. That is their diploid. Males have 16 chromosomes. That is their haploid. Okay. Female produces gametes by meiosis, and male produces gametes by mitosis. As female is diploid on undergoing meiosis, it forms two cells having two haploid cells, which are male, as they have chromosome number 16. And in case of male, it undergoes mitosis, hence 16 goes to 2n, that is 32. That hence female is formed. In this system, males produce sperm by mitosis. They do not have a father. That is, as males, we know female produces male. Here we saw that males can't give rise to males. We saw that too. So they males do not have father, and they also cannot have son. So they have daughter. Okay, but they have grandchildren and grandfather. These female, we know female gave rise to male in honeybees, and male gave rise to female. So these female produced by this male will have another male. Therefore, this is grandfather and this is grandchild which will be produced. An offspring formed from the union of sperm and an egg develops to form as female, which is also known as queen or worker bee. An unfertilized egg develop as a male drone. It is the process is called parthenogenesis. We have studied that parthenogenesis is a development of an organism without fertilization in second chapter, that is reproduction in flowering plants. So this is for today. Thank you.